Hi everyone, I'm going to go over the secondary stains lab in this video. So we are going to talk about the acid-fast stain, endospore stains, both crystal violet and Schoeffer Fulton, flagella stain, and the capsule stain. To give some background, secondary stains are important, especially for bacteria where a gram stain doesn't work, as well as if we want to stain specific structures. So if you want to look at the endospores of bacteria, flagella, capsules, we use these different staining mechanisms. And we're going to go over the acid-fast stain, which uses the Ziehl-Neelsen technique to do the staining. Endospore stains, we're going to talk about two different stains that both stain the endospore, the crystal violet method and the Schoeffer Fulton method, the flagella stain, which uses Leibson's technique, and the capsule stain, which is a type of negative stain. And also crystal violet is also a type of negative stain. Okay, to start off with the acid-fast stain. The acid-fast stain is a differential stain, meaning that it allows us to differentiate acid-fast bacteria from non-acid-fast bacteria. That's what we term a differential stain. There's many different types of acid-fast bacteria, but two important species in medicine are Mycobacterium species of bacteria and Nocardia species of bacteria. So when healthcare suspect that someone has an infection with a Mycobacterium infection or a Nocardia infection, they will send it off to the lab, and the lab will typically do an acid-fast stain. Examples of different infections that are caused by these bacteria. So Mycobacterium tuberculosis, that bacterium causes the disease tuberculosis, which is a bad lung infection. Another type of bacteria is Mycobacterium leprae, which causes leprosy, which is a bad skin disease where you see all these bumps forming, and it's caused by this bacteria. Both of these are acid-fast positive. And then Nocardia causes many different infections that include brain, lung, skin infections. And so when a doctor suspects any of these, the lab will typically do an acid-fast stain. For the acid-fast acid-fast stain is used with bacteria that have mycolic acid in their cell wall. Mycolic acid is a waxy lipid, and so if we look at this image, mycolic acid doesn't let a lot of stains in. That's why we cannot do a gram stain with acid-fast bacteria, because of this mycolic acid. This mycolic acid is good for the bacteria that have it because it makes them much more resistant to heat, to drying, to chemicals such as antibiotics. So bacteria that have mycolic acid are harder to treat than other bacteria. So in their cell wall, they have this very fatty material. And so we have to do the special stain called an acid-fast stain, whereas the gram stain would not work on acid-fast bacteria. For the acid-fast stain, the technique that we do in lab is called the Ziehl-Neelsen technique, and it's the staining procedure so that we can differentiate acid-fast bacteria from non-acid-fast bacteria. The way this staining works, a little bit similar to the gram stain in the steps, is we have a primary stain that's a hot pink primary stain, which is carbofuscin, and this stain will stain everything. And then we add the mordant. The mordant helps the stain stick more, so now all the cells are pink. And then we decolorize with acid alcohol. So we remove the stain from non-acid-fast bacteria that didn't take it in. So acid-fast bacteria at this step after decolorizing are pink. Non-acid-fast bacteria are transparent, and that's where we add our counter stain so that we can stain our transparent non-acid-fast bacteria blue. So in the end of this, our acid-fast bacteria are pink and our non-acid-fast bacteria are blue. The primary stain is the carbofuscin. The mordant is heat that helps the pink stick longer. The decolorizer is acid alcohol, and the counter stain is methylene blue. And this is what our slides look like. So these are various slides where we've done the acid-fast or Ziehl-Neelsen technique on. So if we look at this slide, these pink rods are acid-fast positive, and these are specifically Mycobacterium smegmatis bacteria. These are the bacteria that we have. So in this slide, and this slide, they're all from the same bacteria. It was a mixture of Mycobacterium smegmatis bacteria, which are acid-fast positive, so they are pink rods, and Staphylococcus epidermis bacteria, which are round coccus bacteria, which are blue because they are non-acid-fast stained. So know the bacterial species stained in these slides and that this is the Ziehl-Neelsen technique. The next secondary stain we're going to talk about is the endospore stain. The endospore stain is a special stain that we use to look at endospores in bacteria. So some bacteria form these structures called endospores, which are resting dormant forms of the bacterial cells that the bacteria create when the environment is very stressful. So when there's no nutrients, there's harsh chemicals, there's a lot of dry heat, some bacteria can create endospores, and we see these in Bacillus and Clostridium bacteria as examples. There's a lot more bacteria that make endospores, but these are examples. And again, the endospores are resistant to so many different things. So that's why in hospitals, they really make sure to sterilize everything to get rid of endospores. The two lab techniques to stain bacterial endospores are the crystal violet method, which is a negative staining method, and the Schoeffer-Fulton method. The crystal violet staining method is a negative staining technique, meaning you stain the background. That's what negative staining means. And the crystal violet will stain vegetative cells. The endospores are colorless. So all these colored spots are the vegetative cells that can reproduce. And then the endospores are the white spots or clear spots. So they're the clear ovals inside the purple rods, or they can be seen as free structures. So these are all Bacillus subtilis endospores that were stained using the crystal violet endospore method. Another staining method that we can use to stain endospores is Schoeffer-Fulton. Schoeffer-Fulton is a little bit more involved, and with this method, we could stain the vegetative cells and we could stain the endospores on their own. So in the Schoeffer-Fulton method, our primary stain, the stain that will stain everything, is a malachite green. This is a primary stain. Then the mordant that helps the stain stick even more, or the dye stick more, is heat. Then we decolorize with water, and the primary stain will stick onto the bacterial endospores. And then we add a counter stain to stain vegetative cells, and the counter stain we add is safranin. So this is a result of what Schoeffer-Fulton endospore stains look like. This is a more realistic slide of what the picture looks like. So these slides have Bacillus subtilis, and Bacillus subtilis is a bacterium that forms endospores. The green spots, which look somewhat bluish, but they are green, are endospores, and then the red are the vegetative cells, so the cells that are reproducing, which also can have endospores in them. Next stain we're going to talk about is the flagella stain. In lab, the flagella stain uses Leifson's technique. So flagella is a specific structure that some bacteria have, and it makes them the bacteria that have flagella have an advantage over those that don't, because they can move around. And motility for bacteria is a great thing, because you can swim towards chemicals that benefit you and swim away from things that don't, which is what we call chemotaxis. So moving towards things that are beneficial to the bacteria and away from those that are not. So flagella are these little protrusions outside the bacteria, and we can stain them using Leifson's technique, which stains flagella. The way this technique works is we use a rolling drop slide, meaning you put a little bacteria at one end of the slide and you roll it down a little so you can have it towards the end. Then you let your slide with your bacteria air dry. There's no heat fixing in the Leifson's technique, because heat fixing would harm the flagella, and the flagella are very fragile. And then we add Leifson's flagella stain, which is a mordant that sticks to the bacterial flagella. So this is actually a good slide of what it looks like. So in these slides, we stain the bacteria Proteus vulgaris. Proteus vulgaris has flagella. It is motile bacteria, and it has flagella all around. It's paratrichous. That's what flagella all around is. So here's Leifson's technique, and it's stained the flagella all around. So the mordant sticks to the flagella. And finally, the final stain we're going to talk about in secondary stains is the capsule stain. So some bacteria have an additional layer of protection called a capsule. A capsule is an external layer outside the cell membrane, and it's this gelatinous material that allows bacteria to stick to each other, as well as protecting them from dryness, because dryness can be a form of harming bacteria. So bacteria that make capsules are very hard to treat. They take very tough antibiotics to treat these because of this extra layer of protection they have. And the way we can visualize if bacteria have a capsule or not is by doing a negative stain. So again, a negative stain stains the background. And with this capsule negative stain, we use India ink, which is this really dark dye, to stain the background. And then the saffronin we use as a counter stain to stain the bacterial cells, so that you see this little halo capsule between the bacterial cell and the background. And in these images, we stained the bacteria Klebsiella pneumoniae. And in these images, it's a negative stain, so the background is dark, and the clear capsules are the capsule structures of the Klebsiella bacteria. So this is a capsule stain. And here's a secondary stain chart from your lab manual that's very helpful, that tells you about everything regarding each stain. So I want everyone to study this chart from your lab manual. So know the details of the structure, what everything stains. So for example, the acid-fast stain stains mycolic acid in the cell wall of bacteria. The stain itself, what the technique is called and how it works, and then the bacteria from the images. So I want you to know how you can differentiate a rod from a coccus if I give you a slide picture. So hopefully everyone can do that. Know what the slides look like from the PowerPoint. And let me go back a little. Know the details of the organism. So for example, with the endospore stain, we were looking at Bacillus subtilis. Know the techniques involved. Leaf sense is flagella stain. What's Schofer Fulton? Where's Zeal Nelson? Know all of that, and know which ones are negative stains. So go through everything and understand everything in this chart, as well as all the pictures involved.